Hi students, today we have a discussion with the transcription process, another aspect of molecular genetics. Now this is the first stage in the gene expression process. It is the first stage leading to the gene expression. The second stage only we have the translation process, we will discuss. Now, what did the molecular functioning of the gene reveal? What did the molecular functioning of the gene reveal? So the molecular functioning of the gene reveal what is called the central dogma. The molecular functioning of the gene reveal the central dogma. So the central dogma was proposed by Francis Crick. What is central dogma? The central story of the cell. The central story of the cell. What is happening inside the cell? The secret of the cell. That is called central dogma. That concept was proposed, that observed and discovered by Francis Crick. Now, he just actually observed what is called the central dogma in molecular biology. What's happening? So, the molecular functioning of the gene revealed the replication of DNA, which undergoes a process what is known as transcription. So, this encyclic referring to what is called the replication of the DNA. It is a regular process during the cell division. And this DNA undergoes a process what is called transcription, resulting in the formation of RNA. This is nothing but the copying of RNA on a DNA template strand. Copying of RNA on a DNA template strand is called transcription. We will discuss. So once RNA is formed, so now the flow of genetic information normally to be expressed as a protein in the form of enzymes or any other substance. Now the RNA undergoes translation. The genetic information or the message that is transferred to RNA undergoes translation to form a specific protein. This concept is called what is known as central dogma. So it reveals the flow of genetic information from DNA to RNA, that is by means of transcription process. Then the RNA having the message undergoes translation to form the protein. So this is what we have actually, that is called transcription, what is called the central dogma. Normally, the process of transcription is brought about by a DNA dependent RNA polymerase, because here the synthesis is nothing but the RNA. So the RNA is being synthesized by an enzyme what is called transcriptase. This is an enzyme, transcriptase. This is nothing but actually DNA dependent RNA polymerase. DNA dependent RNA polymerase, the one responsible for the synthesis of RNA. So transcription is brought about by the transcriptase enzyme, which is working under the influence of DNA. That's why I mentioned about DNA dependent RNA polymerase. The elongation of the RNA is brought about by polymerase, as in the case of replication process where we have the DNA polymerase. So this is what is happening. So the process is brought about by, that is transcriptase, functionally it is called transcriptase. The original name DNA dependent RNA polymerase, as it works under the influence of DNA. In some viruses, say an example of retroviruses, or example HIV, or wound tumor viruses. Now the process of transcription is reversed. These viruses have RNA as a genetic material. Now this is RNA which synthesizes DNA. So now RNA directs the synthesis of DNA. Then the DNA undergoes transcription. So this is transcription to form RNA. Then we have the original process, what we have, the translation process. Translation to form the protein. This is what is happening. But you see that these viruses have RNA as a genetic material. The RNA directs the reverse transcription. Then the RNA, which is formed after transcription, undergo translation to form protein. So here you see that one, the process of transcription is reversed. Here you see that one, RNA directs synthesis of DNA. This process is called reverse transcription. Reverse transcription. So this process is called reverse transcription. The enzyme which brings about the process of reverse transcription is called reverse transcriptase. Reverse transcriptase. So this is nothing but, you see that one, what is reverse transcriptase? This is nothing but RNA dependent DNA polymerase. RNA dependent DNA polymerase. See, RNA dependent DNA polymerase, because it is working under the control of RNA. So functionally reverse transcriptase, but originally it is nothing but RNA dependent DNA polymerase. So because the product form is called actually the DNA, we need DNA polymerase only for the synthesis of DNA. That's why I mentioned about DNA polymerase. But it's working under the control of RNA, hence called RNA dependent DNA polymerase. So you can see this one in the case of what is called, that is, retroviruses like XII and also wounded tumor viruses. Now the process of reverse transcription that is observed in the case of tumor viruses is called terminism. So terminism is nothing but what is called reverse transcription. Reverse transcription. The another name for reverse transcription, after the name of the person Temin. He observed reverse transcription in the case of wounded tumor viruses. So that's why it's called terminism. So it's one of the entrance questions also. Terminism is nothing but reverse transcription, observed in the case of wounded tumor viruses by Temin. So we have transcription and another one reverse transcription. Now let us see what are the facts related to transcription process. Number one, for the cell to operate, now the gene must be expressed. Then only the products will be formed. This is number one. This means that whether RNA or protein must be made. That is the nature of the gene expression. When gene is expressed, the product form is either RNA or protein. Now we have different types of RNA. I'll talk about later. Now, one RNA, what is called messenger RNA. One RNA called messenger RNA. So the RNA which carries the genetic information encoding for a protein, that is called what is known as the messenger RNA. The RNA which carries genetic information encoding for a protein from a gene to the cell is called messenger RNA. So it is acting as a template responsible for transferring the information which is later translated to form the protein. Now, for the gene to be transcribed, a gene is nothing but a segment of DNA, you know that one. The two strands of DNA must be pulled apart. Then only we can have the synthesis of what is called RNA. For the transcription to take place, we have the two strands of DNA must be pulled apart so that we can have what is called the hole or what is called a bubble for starting the transcription process. What's happening? The RNA polymerase, see that one, the RNA polymerase binds to the DNA segment. And you see that what, as a result, binds to the DNA segment, where at the start of the structural gene, the one which is going to be transcribed at the start of a gene, so that what is happening, the double helix being opened. So at the start of a gene, the RNA polymerase binds to the DNA segment so that we are getting a transcription bubble, the starting point, and as a result the RNA will be synthesized with the help of RNA polymerase. Now the RNA is synthesized by RNA polymerase, I mentioned about here. Now the nucleotide sequence in RNA is complementary to the nucleotide sequence of template strand of DNA. The sequence of what is called the nucleotide sequence of RNA, which is normally synthesized, is complementary to the nucleotide sequence of DNA strand, which is acting as the template from which the RNA is synthesized. This is about a few words about this one, what is happening during the process of transcription. Now we have, so why both the strands are not copied? Why both the strands are not transcribed or not copied to produce mRNA? We have to see the reasons for that one. During transcription, only one strand is acting as a template and another strand is being normally displaced. So both the strands are not acting as a template simultaneously to produce RNAs. Why? Let's see. Why are the two strands of DNA not copied simultaneously? There are two reasons. Reason number one, if both the strands act as a template simultaneously, it would result in the formation of two RNA molecules and each with different sequence of nucleotides. Number one, so two RNA molecules with a different sequence of nucleotides would result. As a result, what will happen? It leads to, that is, the formation or translation of two different proteins, because RNA is encoding for two different proteins with a different sequence of amino acids would result. Two different proteins with different sequence of amino acids would result. That will affect normally the genetic information transfer machinery. So two RNA would result. That would result in two different proteins with a different sequence of amino acids, and that would affect the genetic information transfer machinery. Number two, if two RNA molecules are produced, because the two RNA molecules are complementary to each other, there is a chance of getting a double-stranded RNA model or a structure, double-stranded RNA structure, and each strand is complementary to each other. Because of this double-stranded RNA, it would prevent the RNA being translated to form a protein. So double-stranded RNA helix form, that will prevent the RNA being translated into protein. So the translation is affected. The first one, we have the genetic transfer machinery is affected because of the formation of two molecules of RNA, which would result in the formation of two proteins. The second is formation of two RNA molecules, and which are complementary to each other. There is a chance of having or getting what we have, a double-stranded RNA molecule. This double-stranded RNA molecule will not be translated to form a protein. So these are the two reasons why both the strands of DNA are not not actually transcribed, not acting as a template simultaneously. Now let us go to the next part, the transcription unit. Now what are the components of the transcription unit? So the transcription unit is determined by three components. Component number one, that is a promoter. Component number two, the structural genes. And component number three, the terminator. These are the three components present in a transcription unit. Now what is a promoter? Where is it located? The promoter is a segment of DNA which is located towards the 5' end of the coding strand. I'll tell you what I mean by coding strand later. So the location of promoter, we can say either way, but I am saying only one way. a sequence of DNA which is normally formed towards the 5' end of the coding strand. Or also we can say towards the 3' end of the template strand. So 5' end of the coding strand or 3' end of what is called template strand. So we take only one towards the 5' end of the coding strand. Now what is its function? It is a recognition site for RNA polymerase to initiate the transcription process. Now the RNA polymerase responsible for synthesis of RNA must bind to the promoter. It is the one which directs the RNA polymerase towards the transcription initiation site. Now this is what we have, the transcription initiation site, where we have the structural genes are present. So the RNA polymerase binds to what is called the promoter region. This is the binding site for RNA polymerase, because RNA polymerase cannot find the transcription initiation site. It has to be directed with the help of somebody. The somebody is nothing but what we call the promoter. Simply we can say promoter is a region to which the RNA polymerase binds. It is a recognition site for RNA polymerase, present towards the 5' end of the coding strand. The second one, what we call the structural genes. They have the message, they have the codes for the transcription, so that we have the protein will be formed. It may be one gene or many genes, we can discuss later. So the second component, the structural genes. Normally the promoter is present next to the structural gene in the upstream, towards the left side. This is called upstream. Towards right side is called downstream. So it is present towards upstream, next to what we have, that is structural genes. They are responsible for the transcription to form or that is a protein or enzymes. Now the third component, what we call the terminator. It is present towards the 3' end of the coding strand. This is towards the 5' end of the coding strand, and this one terminator is present towards the 3' end of the coding strand. Once the transcription is going to be completed, now with the help of RNA polymerase, the sequence of DNA just towards the 3' end stops the RNA polymerase from transcribing the RNA. So it stops the transcription process by preventing the RNA polymerase to transcribe an RNA. That is a terminator, nothing but a sequence of DNA. So all the sequence of DNA only, either promoter or structural genes or terminators are actually, that is, units present in the DNA concerned with the transcription process. Now within promoter, there is a specific site to which only the RNA polymerase binds, and that is a site which directs the RNA polymerase to the transcription initiation site. What is the name of that actually site within the promoter? That is called Tata box. Tata box. It's also called Goldberg Holmes box, after the name of the two persons, Goldberg Holmes box. So what is Tata box? A sequence of 6 nucleotides, for example TE, 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 sequence of 6 nucleotides, and they are responsible for being recognized by the RNA polymerase. RNA polymerase recognizes a promoter, that is a specific region within the promoter, what's called Tata box. It is a Tata box, that is the one which directs the RNA polymerase to the site of transcription. That's the function of that one. So in eukaryotes, it is called Tata box. In the case of prokaryotes, it is called Grignard box. In the case of, this is in the case of prokaryotes, the Tata box. This is in the case of eukaryotes. This is in the case of, that is what we have, the prokaryotes. Prokaryotes. So in addition to these 3 units, the promoter, that's sorry, the structural genes and terminator region, we have what is called enhances. What are enhances? What are enhances? These are additional regulatory sequences. Additional regulatory sequences found beyond the promoter, helping in transcription. So enhances also recorded in the case of eukaryotes, not in the case of prokaryotes. These are all additional regulatory substances found beyond the promoter. This is left side. We have also silencers, that is enhances, insulators. These are all the different types of additional regulatory substances found beyond the promoter, helping in the transcription process. Now, we have to go for the next one. What is a template strand? What is a coding strand? Now, the DNA of the structural genes. Now, this is an area. The DNA of the structural genes. That is the two strands of DNA of structure. The two strands of DNA in the region of structural genes of the transcription unit have antipolarity. The two strands of DNA, the structural gene region of the transcription unit have opposite polarity. One is running in 3-5' direction, another one is running in 5-3' direction. Normally, either replication process or transcription process, it always occurs in 5-3' direction. 5-3' direction. Replication or transcription always occurs in 5-3' direction. The strand, which is acting as the template for the synthesis of what is called RNA, is called as a template strand, which is having the polarity 3-5'. So the strand having the polarity 3-5' is acting as a template strand for the synthesis of mRNA. So here only we have the formation of RNA. The strand having the polarity, that is 3-5', which acted as what is called a template for the synthesis of RNA, is called template strand. So its polarity is 3-5'. Now that is why we have the RNA being synthesized in 5-3' direction, because the two strands, that is RNA, the template strand, are complementary to each other. Suppose, for example, I am taking this is RNA. So it is starting with 5-3'. Now the transcription always occurs in the direction of 5-3'. Now the RNA and the template strand are complementary to each other, except that here we have normally thymine, but here we have uracil. Suppose we have thymine here, in place of thymine we have uracil. That's the only difference between what we have, the template strand and the RNA. The mRNA which is being synthesized, both are complementary to each other. In the case of template strand we have thymine, where in the case of RNA we have uracil. Now, another strand which is being displaced. So this is the coding strand, the original strand having the message, but which will not be transcribed. If it will be transcribed, then we are not getting what is called actually the copy of mRNA. In a different way normally we are getting. Now the strand which is having the polarity 5-3', that is normally, which is complementary. So this is complementary to each other, that is the template strand is complementary to RNA, whereas the coding strand is similar to the RNA strand, except that here we have in the coding strand thymine is present, whereas in the RNA, that is uracil is present. See, the template strand, the synthesized RNA are complementary to each other. The meaning for that one, if you have just a T here, then it is forming a pair with what is called adenine. These two are complementary. Okay, in RNA uracil is present, again thymine is present. But here the coding strand is similar to RNA, not complementary, similar to RNA, except that it has thymine instead of uracil. That's the only difference between the coding strand and RNA. So coding strand and RNA are similar, except your acid and that. Whereas the template strand is complementary to the RNA. Okay, this is one. So coding strand is displaced during transcription. The meaning for that one, it is not taking part in the process of transcription. That's about what is a coding strand. It is the original strand having the information, but it's not being transcribed. If it is transcribed, we are getting what is called not a similar RNA similar to coding strand. That's why another strand is acting as a template or a model for the synthesis of RNA. Benzer in 1955 proposed the concept of recon, muton and cistron, all referring to the segments of DNA. What is recon? A segment of DNA which undergone recombination. What is muton? A segment of DNA which undergone mutation. What is cistron? A functional unit of DNA, a segment of DNA that specifies a protein or polypeptide. That is called cistron. Now, if we are taking the gene, there are two different types of genes. One, monocystonic gene, another one polycystonic gene. Likewise, also we can say monocystonic mRNA, polycystonic mRNA. A monocystonic gene is found in the case of eukaryotes, including human beings. The meaning for that one, there is only one cistron that encodes for one protein that also being transcribed into one mRNA. So, there is one cistron which is normally undergoing transcription process to form a single mRNA encoding for one protein. When translated, it forms a single protein. So this is called monocystonic mRNA, having a gene encoding for a single protein or polypeptide. It is a characteristic feature of all eukaryotes. But in the case of human being, the hemoglobin gene is polycystonic. There are 4 cistrons, 2 alpha, 2 beta. Each one is specifying one polypeptide. Because in hemoglobin we have 4 polypeptide chains. That's an exception. Now what do you mean by polycystonic gene? Here there are many cistrons in the structural genes, many cistrons. And all the cistrons are transcribed to form only a single mRNA. Here also only single mRNA, here also only single mRNA. Though we have only one cistron here, there are many cistrons. The single mRNA encoding for many proteins being translated to form many proteins. So, many cistrons in the structural genes, but only one mRNA with many coding genes, that is many cistrons coding for many proteins. It's very common in the case of prokaryotes. Polycystonic gene, many cistrons transcribed to form only single mRNA with many, that is coding genes, resulting in the formation of many proteins. So there is a combination of monocystonic and polycystonic gene. So in the case of monocystonic gene, what I mentioned as per the book, we have, there is one gene which is encoding for one mRNA, which contains one gene, mRNA gene, coding for only one protein. In the case of polycystonic gene, there are many genes, a cluster of genes together, present on the chromosomes. They are all actually just transcribed together as one unit in the form one mRNA, being translated into many proteins. So, one cistron, one mRNA, one protein. Then many cistrons, but only one mRNA, many proteins. That's the result. So, or we can say also, a single promoter directs the transcription of one cistron to form one mRNA, which is being translated from a single protein. Again here, a single promoter directs the transcription of many cistrons of structural genes into a single mRNA having many genes encoding for many proteins, which are actually cluster of genes inherited together to form many proteins. That's the meaning. Okay, so one cistron, one mRNA, one protein. many cisterns but only one mRNA coding for many proteins, resulting in many proteins. That's about what we have monocystonic and polycystonic gene. These genes were reported by Richard J. Robert and Philip A. Shaw. They only just proposed, formed the monocystonic gene and polycystonic gene. Don't forget, monocystonic gene is found in eukaryotes including humans, and polycystonic gene is found in the case of prokaryotes, that is bacteria. Before starting the transcription process, now the RNA polymerase would bind with a recognition site in the promoter region. If you are taking the RNA polymerase in the case of prokaryotes, it is formed of two components. Component number one, the core enzyme. This is the component number one. Component number two, the sigma subunit. Now the core enzyme is actually formed of many subunits. As per the book, it is given B, beta 1 and alpha. But normally speaking, we have 5 subunits. That is alpha, alpha 1, beta, beta 1 and omega. But in our textbook, it is given just beta, beta 1, alpha. That's the core enzyme components. Now what is the role of core enzyme? Polymerization of nucleotides for mRNA. That is nothing but the synthesis of mRNA. There is a role played by the core enzyme. The core enzyme cannot initiate the transcription process. It needs an initiation, what is called a factor. That initiation factor is a sigma subunit associated with what we call the core enzyme. After initiating the process of transcription, it may be left out. Okay, so it is called as an initiation factor for transcription, or it is for recognizing the site in the promoter region for the binding of RNA polymerase. Now the promoter is varied from just one organism to another. What is the role of RNA polymerase? It opens up the DNA double strand, producing what is called a transcription bubble. Suppose this is the DNA. This is the region what we call the formation of a bubble, transcription bubble. That means that this is what's called the, I will talk about more. Now this is called transcription bubble. This is because of the separation of the two strands by the RNA polymerase. Now the RNA polymerase synthesizes or manufactures RNA by adding, that is the nucleotides one after another ahead, leaving the sigma factor outside. Now this is the RNA which is being synthesized, and this is what we have, the RNA polymerase which leaves this RNA. Now this is the new RNA that is being synthesized. Now this is the sigma factor which is being left out in the promoter region. We'll talk about data later now. So while the process is going on, the nucleotides are added for manufacturing the RNA so that the RNA is growing. Once the function of the initiation factor, the sigma factor is over, it is being left in the promoter region. Now we have actually what's happening at the end of the structural gene. We have a terminator sequence. The terminator sequence stops the process of what is called transcription. Now the terminator sequence actually is that when RNA polymerase produces, the terminator sequence forms the RNA as a hairpin-like structure. The terminator sequence forms the RNA as a hairpin-like structure. Now we have a subclass of terminators have a recognition protein to function. The termination factor, it may be intra-DNL structure or extra-DNL structure. Now the subclass of terminators recognizes a protein called the Rho protein. This is what's called the Rho protein. It's a termination factor. We'll discuss in detail. What's happening here in a nutshell, only I am saying that one. So now the sigma factor is left. Now the addition of one by one the nucleotides to the growing RNA, once it reaches the terminator region at the end of the structural gene, now the process of transcription stops. This is because of some functional protein, what's called the Rho protein, which is responsible for relieving the nascent, that is RNA polymer, nascent, that is RNA. Nascent means the newly formed RNA. So with the help of Rho protein, now the RNA, which is completely synthesized, is being actually removed from the DNA. So this is what is happening. I am giving only natural. What is the role of this RNA polymerase? Responsible for actually the initiation process as well as what is called the polymerization process. It utilizes that energy in the form of what is called adenosine triphosphate. So once it reaches the termination region, the RNA polymerase, the terminator sequence stops the transcription process and forms RNA as a hairpin-like structure. Then the subclass of terminators have a recognition protein and they terminate the process of transcription, and so that the completely formed RNA being relieved along with RNA polymerase from the DNA.